Thank you, everyone. Doug was kind enough to give me and Boris some of his time today because we had such a long presentation. So thank you, Doug. And he'll be presenting some of his innovative methods at a poster tomorrow, looking at these same types of data. So I know all of you are familiar with Mommy's scientific goals to define the roles of maternal fetal immunity using different de novo and recall vaccines, as well as new vaccines that have become available during the Mommy time period, and viral infections and pregnancy complications to understand more about the maternal fetal dyad and how immunity gets transferred across that dyad. We have two projects that are supported by the data management core, a clinical core with many bank samples, and the administrative core. We had some changes in our investigators this year. Unfortunately for us, fortunately for her, Michal Elevitz left Mount Sinai School of Medicine to become the CEO of a private foundation. So she's no longer going to be one of the MPIs. And then we welcome back Galit Alter from her sabbatical at Moderna, who is going to co-lead Project 2 with Boris Yulk, and also Chloe Villani from the Broad Institute and MGH is joining our Project 1 team to work with us on our single-cell sequencing data. We are fortunate to work with many outstanding postdoctoral associates and staff scientists, many of whom are here at this meeting, and one of whom was not able to come due to visa issues, but she will be presenting her flash talk on Zoom, Racheli Normand, and it's also her birthday today, so if she's watching, happy birthday, Racheli. The goal of Project 1, you're all familiar with from prior presentations. So some of the work that we present in this session will focus on parts of AIM-1 that overlap with Project 2, defining maternal single-cell immune profile in response to the de novo COVID-19 vaccine across trimesters and in comparison to non-pregnant individuals' response to that vaccine. And I'm going to be focusing on AIM-3, defining placental signatures associated with immune-mediated pregnancy complications. And some of this work will overlap very nicely with our IOF that I'm presenting tomorrow about spatial transcriptomics in the same individuals. So the placental single-cell RNA-seq we carried out this year in collaboration with Chloe Villani's lab, and we have two amazing data scientists and computational biologists working on this, Dr. Andy Lopez and Dr. Racheli Normand. This is our cohort, 70 individuals. We have 3 different control groups. We have women who underwent labor and whose placentas were collected at term. We have planned C-sections at term, as well as planned C-sections preterm, who can serve as controls for our preterm pregnancy complications. Our 5 pregnancy complications that are immune-mediated of interest include type 1 diabetes, fetal growth restriction, preterm preeclampsia, term preeclampsia, and preterm birth. And so we have fresh placenta collected from delivery that is either cryopreserved and made into a single-cell suspension, on which we carried out 10x multimodal single-cell RNA-seq, site-seq, and TCR-seq, or spatial transcriptomics, which I'll tell you more about tomorrow. This table shows the ends that we have for either our spatial transcriptomics on the left, the tissue microarray, or our single-cell RNA sequencing results that I'm presenting today in this session on the right. We excluded patients who had either chronic or acute inflammatory conditions that might influence their placental signature, as well as people on immunomodulating medications. Before we went forward and did the single-cell RNA sequencing on 70 placentas, we did a pilot to examine whether we wanted to use single nucleus versus single-cell RNA-seq, and also piloting different kinds of preparations for the single cell. So I know this is somewhat of a technical group, so in case this is of interest to you, just a brief note about, we found that single nucleus really only had good representation of trophoblast populations, and we really were losing all of these immune cell populations with the single nucleus preparations. And this is consistent with some other groups, like a HubMap group that I'm in touch with, who is finding the same. So we made the decision to really proceed with single cell, and we looked at different kinds of preparation, like a debris removal and dead cell preparation, a cell sorting preparation, and a Percol gradient. And ultimately, we decided to proceed with the 75-20% Percol gradient to remove dead cells, debris, and red cells, and permit us to capture all of these other immune cell populations, as well as our trophoblast populations of interest. You can see here that we ended up with a great number of high quality cells per sample that was relatively uniform across the conditions. Some of the very high number of cells were excluded if they were outliers. And we ended up in only the first 50 samples with over 1 million high quality placental cells to analyze, making this the largest and most comprehensive immune cell placental atlas of pregnancy. And you can see that we get a lot of resolution on different cell types in the placenta. So we have 3 different stromal populations, and these are sort of the parent lineages. I'm going to show you some subclustering that we've done that's more granular of each of these lineages. But we also have vascular endothelial cells, platelets, 2 different large trophoblast populations, extravillous trophoblasts and syncytial and cytotrophoblasts. And then we have fetal placental macrophages or Hofbauer cells, which we found play a very important role in many of these pregnancy complications, maternal resident myeloid cells and monocytes, plasma and B cells, and T and natural killer cells.